Hi guys, so this video is all about the triple sugar iron agar test, or what we call TSI. The triple sugar iron agar test is one of the biochemical tests used to differentiate and identify gram-negative bacilli. So triple sugar iron agar test is used for the identification of enteric bacteria or Enterobacteriaceae. It is also used to distinguish the Enterobacteriaceae from other gram-negative intestinal bacilli by their ability to catabolize glucose, lactose, or sucrose, and to liberate sulfides from ferrous ammonium sulfate or sodium thiosulfate. So this one is our negative control. This is an uninoculated TSI agar. And we have the slant part. This one is the slant part. And this one is what we call the butt part. So TSI slants contain 1% of lactose and sucrose and a 0.1% glucose concentration. This medium also is incorporated with phenol red, a pH indicator that turns yellow when there is acid production from carbohydrate fermentation. So how do we inoculate a TSI agar? So you have to get your bacteria from your culture using an inoculating needle, and then what we have to do is to stab the agar dip straight. So this one, stab the agar, and then using a zigzag pattern streak the surface of the slant. This one. Then cover your tubes. After that, you have to incubate it for 18 to 24 hours in order to detect the presence of sugar fermentation, gas production, and hydrogen sulfide production. There are different reactions that may occur using the TSI agar tube. So first we have the K over A. So this one is K over A. So the numerator represents the slant part, while the denominator represents the butt part. K is for alkaline and A is for acid. Remember, the pH indicator phenol red turns yellow when there is acid production. So this reaction is due to the fermentation of glucose, in which phenol red indicator turns yellow due to the increased acid or due to the persistence of acid formation in the butt. The slant remains red or alkaline because of the limited glucose in the medium. Remember, it only contains a 0.1% glucose concentration. Therefore, limited acid formation which does not persist. That's why it remains alkaline. So next we have the A over A. Both are yellow, slant and butt, so it is written as acid over acid or yellow over yellow. This is due to the fermentation of lactose and sucrose. So meaning the bacteria was able to ferment lactose and or sucrose. That's why the slant turned yellow, and due to the high concentration of these sugars in the butt, so the butt also turned yellow, leading to excessive formation in the entire medium. So this is A over A. Next we have gas formation. So this is also A over A. The slant and butt are both acid, that's why A over A. But as you can notice, there is splitting of the agar, indicating gas formation. So this is This is written as A over Ag. G stands for gas production. We also have the hydrogen sulfide gas formation, which is seen by the blackening of the agar. A red dot and slant indicates that none of these sugars were fermented, neither gas nor hydrogen sulfide were produced. So again, let us interpret the result. So when we have A over A, it means that glucose and sucrose and or lactose are fermented. For this one, we have A over Ag, meaning glucose was fermented with gas production. Sucrose and or lactose are also fermented. K over A, meaning that glucose was the only sugar fermented. And K over A plus H2S, meaning glucose was fermented and hydrogen sulfide was produced. For the K over K, meaning no sugars were fermented. So that's how we perform the triple sugar iron agar test, and that's how to interpret and report the results. Thank you.